how does the brain interpret what we see? The journey of light. Our vision begins with light entering the eye, but the process that allows us to experience a coherent world is remarkably complex. Light reflects off objects, passes through the cornea and lens, and reaches the retina at the back of the eye. The retina contains millions of specialized cells, including rods for low-light vision and cones for color and fine detail. From light to signal. These cells convert light into electrical signals through biochemical reactions. The signals travel along the optic nerve toward the brain. What moves forward is not a picture, but coded information representing contrasts, colors, motion, and brightness. The optic chiasm. At the optic chiasm, fibers from each eye partially cross. This arrangement ensures that the right visual field is processed in the left hemisphere and the left field in the right hemisphere. This crossing supports binocular vision and depth perception. The thalamus relay. From there, signals pass to the lateral geniculate nucleus, LGN, in the thalamus. The LGN acts as a relay station, sorting and emphasizing certain elements such as contrast changes and motion cues. It prepares visual information for further analysis in the cortex. The primary visual cortex. The signals then reach the primary visual cortex, V1, in the occipital lobe. In the 1960s, Torsten Wiesel and David Hubel discovered that V1 is organized into columns of cells, each responding to specific features such as edges, lines, or particular orientations. Their work revealed that vision is processed through a hierarchy of increasingly complex stages. Progressive analysis. Cells in V1 detect simple patterns. As information moves to areas like V2, V3, and V4, neurons respond to more complex shapes, textures, and color combinations. Other regions, such as MT, specialize in motion. This layered system allows the brain to build a detailed interpretation of the world from basic elements. Critical periods in development. Hubel and Wiesel also showed that early visual experience is essential. During a critical period shortly after birth, the developing brain must receive appropriate visual input. Without this stimulation, certain neural pathways fail to mature. Their discovery influenced modern treatments for childhood visual disorders such as amblyopia. Higher-level recognition. Higher-level processing continues in the temporal lobe, particularly in the inferotemporal cortex. Here, neurons respond to complex objects such as faces, hands, or familiar items. The fusiform face area, FFA, is especially important for recognizing faces. The parietal lobe, meanwhile, handles spatial awareness, helping us judge distance and interact with our surroundings. The two visual streams. These regions form two major visual streams. The ventral stream, or what pathway, identifies and categorizes objects. The dorsal stream, or where or how pathway, processes location and guides action. Together, they allow us to recognize a cup, while also understanding where it is and how to grasp it. Seeing as interpretation. The brain does not simply record what is in front of us. It interprets, predicts, and sometimes fills in missing details. Visual illusions demonstrate that perception is an active process influenced by memory, expectation, and context. Much of what we see is the brain's best interpretation of available information. Nobel Prize discoveries. In 1981, Hubel and Wiesel were awarded the Nobel Prize in Physiology or Medicine alongside Roger Sperry. Sperry's work on the divided brain showed that the two hemispheres can differ in how they process information, adding another dimension to our understanding of perception. The nature of vision. Together, these discoveries established that seeing is not a passive act. It is a dynamic, developmentally shaped process involving millions of coordinated neurons. Each moment of vision relies on the brain's ability to decode patterns of light and transform them into a rich world of shapes, colors, motion, and meaning.